Welcome to here. Here I will be discussing about the freeze fracture technique. So first of all, we should know what is it and what is its purpose. So it is an experimental approach or technique which gives the structural details of cell plasma membrane. Here I will say the overview of the processes and how it is done. At first, as a process, at first cells are frozen and immobilized with the help of a liquid nitrogen at minus 196 degrees Celsius. Alongside, cryoprotectant is used to prevent distortion from ice crystal formation. Here, cryoprotectant is acting as a anti-freezing agent. Then, cold microtome knife is to be used to fracture the frozen specimen. As a result, bilayer of the membrane will be splitted into monolayer plane. You can, you can just visualize when bilayer will is splitted. Then, mainly we know that the plasma membrane is formed the bilayer. So when this bilayer is splitted, then monolayer plane will be exposed. But the problem is this fractured plane cannot be observed directly. So to solve this problem, shadowing needs to be done by heavy metals such as platinum deposition. Mainly by platinum it is done. And next, for sufficient strength, a carbon layer is then deposited on the top of the metal layer. So this carbon layer impart the strength of this structure, so it will not be fractured or it will not be broken down. Finally, the organic material of the cell is dissolved away to leave only the thin metal replica of the specimen. At last, the metal shadowed replicas are viewed in electron microscopy. Here we can see the specimen is frozen or immobilized on the support. Then in the next step, shadowing is done by heavy metal. Most of the cases the platinum heavy metal is used for shadowing, and you can see here that the gray color on the specimen, and this gray color is the shadowing by heavy metal. And in the next step, to impart the strength and total structure, carbon layer deposition is done. Then at the final step, by removing the support and by dissolving the organic material, only the replica will be left, and this replica will be visualized by electron microscope. Here I just mentioning the steps in the short detailed notes. Like first of all, the cryofixation is done, then fracture the specimen by knife. After that, the heavy metals deposition followed by carbon deposition will be done, and then remove the specimen from the replica, and finally observe the total structure or total replica will be done by transmission electron microscope. So if you like this video, please press the like button and do subscribe.